fact, it's a way that you can track variation in species by looking at duplication events that have happened over evolutionary time. Who here took 241 here at CSU? Were you guys, what was your genotype for the ALU sequence? Remember when you swished around stuff inside your mouth, you spit it out in a cup, you extracted DNA, you ran PCR. Nobody remembers anything. Do you remember the ALU sequences? You guys looked to see if you were homozygote minus, homozygote positive for the inter heterozygote. Yeah. So that's, you're looking at duplication events. So those were showing how you guys, that's called, you've ever seen the term copy number variation. CNVs, your book talks a little bit about them. I don't care that you guys know all the details about copy number variation, but this is the natural, this is what you guys track, those folks who took 241 lab here. You looked at your genotype for an ALU sequence and you guys were all different. So you're showing amongst the human population how we can see big duplication events, okay? And those are fairly long sequences that have been duplicated and either deleted or gains and losses of sequence over time. So duplication's not quite so terrible. What about the relocation one? Is that kind of depends, right? So it depends on where you relocated it from, and we'll get into more detail, but where the insertion point, what kind of control it's under. So if you had a gene that was included in that segment that was under the control of a promoter or some other enhancer sequence, and all of a sudden you throw it over here, you might see different levels of gene expression happening, and that can be bad or maybe have a neutral effect. So that's really dependent. But overall, of those three, gain, loss, and then relocation loss is generally the worst on an organism. Okay, so we're going to look at inversions first. So there's sort of two types of inversions, and they're characterized dependent on whether they span the centromere or whether they're on the other one side or the other from the centromere. This is, again, the reason that this terminology comes into play is based on cytologists, so people that were looking at chromosomes and cells, and they could see across the centromere. If you're looking on a microscope, you could see these changes. So that's why they've been categorized into these two different types. So your first one is called paracentric. And this is where, if you have your chromosome, so this is our normal chromosome here at the top, the wild type. And if it were to undergo a paracentric inversion, what we're saying is the centromere is actually not included in that inversion. It's on the outside of the inversion. So, and it can happen at either end of the chromosome, so it's not regulated to one side or the other. But in this case, it would be your purple and the green segment get flip-flopped. So that's paracentric. So wherever that inversion event is happening, the centromere is not involved in that inversion, it's outside of the inversion. And that's in contrast to peri. So these are always fun ones to memorize. You have paracentric and pericentric. Peri means the flip-flop, that that inversion event is happening spanning the centromere. Okay. So it includes the centromere. Right, so in this case we can have the green and the black segments are flipped to each other. I think my chromosomes are degrading as we go along. It's all like wibbly wobbly. Okay. And what I wanted you guys, so I'm doing this right now in just, color, kind of color segments, but of course if you can picture alleles or gene segments, gene sequences, and you change those, the order of the genes change, the order of the alleles change, all that kind of stuff. The same way when we think about recombination of alleles, okay? And I'm just doing it sort of diagrammatically right now with color blocks, but I would definitely encourage you guys to consider it being, okay, what if we had alleles on here that are now, the order of them is going to be changed, okay, if we change the whole sequence segment. And again, I know we're not going into if you're really excited about chromosomes and you want to know more, we're not going into a ton of detail about how this happens and how your chromosome can fix it. We'll get into a little bit when we get to repair of mutations. But I just wanted you guys, this is a little more of a conceptual, memorize paracentric and barycentric and then regurgitate it onto the page for me type of concept here. For the most part, inversions don't tend to have phenotypic consequences. I'll put usually. And that's because this is a balanced rearrangement. So we're going to talk about when you have unbalanced. But in this case, this is a balanced rearrangement of genetic material. Right? So you're not really losing or gaining major parts of the genes of the DNA. You're kind of keeping all the same DNA. So by balance, there's no gain or loss. But so I wanted you guys to think a little bit, when would it be an issue? When would we expect to see phenotypic consequences?
Small chromosomes have to line up.
Yeah. So if when your homologous chromosomes need to line up for mitosis, depending on how big the sequence is that was flip-flopped, this might be a problem for them finding each other and lining up. When would it be a problem phenotypically? And I'll come back to Carl's comment because it's important for there's a part of that book that I wanted to point out.
If there's something that happens in something vital, they really break.
Yes. Yeah. Yes. Yeah. So it's thinking about where. So I'm just drawing these random colored segments, but where this break occurs and the flip-flop happens, if you break up a gene, if that happens in the middle of a gene, you probably have some pretty serious, it's probably going to be a non-functional amino acid that's now being coded for. So where you have problems, kind of a when or how or why, when, where. And so one is if the break points in a gene. And then the other one is, again, has to do more with the gene expression, regulation stuff, which we haven't quite gotten to yet, but you guys have some knowledge of it from your prerequisite courses. genes are controlled by some on-off switch, right? There's enhancers and silencers, there's parts of the genome that are not protein coding sequences, but they help turn on, turn off, turn up, down genes. So if you move these sequences around, and if that new location is under some other type of gene regulation and control, so if you move a gene, and its new location now puts it in a really silent region of the genome or a highly upregulated region of the genome. Either way, somehow now your gene's under different expression control. So whatever is, abnormal for that gene. As it turns out that where genes are actually matters to a certain extent. It depends on the gene. But the area of the genome where a gene is located usually has sequences around it that are affecting whether the gene is turned on, turned off, and all those different things. So surrounding regions, and you guys are used to thinking about that. You've learned about 5' UTRs and 3' UTRs, untranslated regions that help control gene expression that we're going to get into after second exam. Location of the gene matters. So if you move it to a new region, it might have phenotypic consequences. And then the last piece is what Carl. So do these two items make sense to you guys? Okay. The last thing was what Carl had brought up in terms of, and it's called an inversion loop. And I'll explain why I'm writing it up here. And really what you can write in your notes is, I don't have to know this for the exam. So your book goes in, it's actually really cool stuff, but it's just one of the many genetic topics that we're not covering in this course. But it's when you have this, and as Carl said, if these are trying to come together, it's when you're a heterozygote for this inversion, which means you have one normal chromosome, one inverted chromosome. Now, if you had both normal, they meet up just fine and they line up, their colors all match and everything's happy at mitosis or meiosis, right? If they're both inverted the same, so say these are both flipped here the same, again, they can match up just fine. When you're in the heterozygote condition, so you have one inverted chromosome like this and one normal, when these go to pair, okay, and they're finding, it gets to this region and you have problems, okay, because they don't know how to match up based on homologous sequences, because homologous sequences are out of alignment. What they do, and you can look in your textbook if you're interested, is the one chromosome does this crazy roller coaster loop-to-loop formation, and it still manages to match up just fine because it inverts itself, like it flips its own sequence around so that it'll match up to the normal. So they pair up just fine, but because of that crazy loop structure, when they go to undergo anaphase and pull apart, often the chromosomes break, you have all other kinds of problems that occur. And so when we see phenotypic consequences, maybe not in the individual who has an inversion, but in their gametes. When they go to try to do meiosis, those chromosomes are having problems. So conceptually, you guys can kind of wrap your heads around that more of a basic general point that maybe the individual who has the inversion will have a phenotypic, nothing that shows, but they may tend to have a lot of miscarriages or some other issue because it just can't go through meiosis properly. But there's a whole section in your book that talks about, it's called an inversion loop, okay, and it's describing that crazy chromosome structure. I will not test you guys on this on the exam, but it is some really neat stuff if you are just like, I actually have extra time and I want to read about inversion loops. That was sarcasm. Okay. Make sense? Questions? Okay. Translocations is your next piece. I actually consider sometimes telling you guys, you need to learn all this, go read it, but then not testing you on it. But I decided, I'll just let you know I'm not going to test you on it, and everyone's just going to rip that page out of their textbook, use it to light their little jack-o'-lantern on Friday. Translocation. Okay, so this is where chunks of genetic material from one chromosome are being stuck onto other chromosomes and all crazy willy-nilly. This is really, really common in cancer cells. Cancer cell karyotypes are one of my favorite to look at because it's like, I don't know how to describe it. It's like you're in crazy land, It's like you take your normal karyotype of a human that looks so neat and orderly, and now all of a sudden there's these crazy long chromosomes and crazy tiny short chromosomes and all bits of material are every which way. It's like everything that could possibly go wrong in the cell is wrong in the cell, which is maybe a morbid way to think about it. But I spent a lot of time looking at thousands of chicken tumor cells, so they have lots of things like translocations. And there are many different types, as you can imagine. So all different combinations of ways that genetic material can be moved around. But we are going to focus on one. Look at this. I already told you, don't worry about inversion loops. You only have to know one translocation. And it's the simplest one. So this is called a reciprocal or balanced. Okay. So this means that whatever chunk is relocated onto the chromosome, it's an even exchange. So one piece breaks from one chromosome, gets introduced onto the next chromosome, and that same, so it's a reciprocal exchange, essentially. So each chromosome is going to exchange a piece of their genetic material. So it's balanced. You don't, again, no gain or loss of genetic material. And because it's balanced, again, there's sort of a minimal phenotypic consequence. Although, again, the same as with, so the same concept that we just wrote up here that applies to inversions applies to this reciprocal translocation, where you cut and break off this chromosome to exchange with this chromosome. If you break in the middle of a gene, you're going to have a problem in your phenotype. You'll see something manifest itself in the phenotype. Or again, that whole concept of what happens when these chromosomes come together for mitosis or meiosis. Then we see problems. And again, resulting in the offspring more so than the individual who has the translocation. So when they go to undergo meiosis, you're going to have some problem. There are two ways that we can end up with a reciprocal translocation, or two common ways. So one of the ways is if, and this is what happens really common in cancer cells, is when chromosomes break, and they're non-homologous, so it's two different chromosomes, right? So the whole, in fact, that's a point I should make. We've talked about crossovers and recombination happening at meiosis, right? Homologous chromosomes together, they come together, exchange genetic material. That's not a translocation. Okay? That's normal in the sense of genetic material exchange. So two chromosomes exchanging material, that's a pretty normal activity. when they're homologous chromosomes. So this is a key kind of point that if you're talking about different chromosomes, non-homologous chromosomes exchanging material, that's when we consider it a translocation. That's when it stands out as not wild type, an abnormal situation. Okay. So if they break, and this is a double-stranded break, and as we get into mutations and repair, we'll be talking about double-stranded breaks a lot, so my shorthand for that is going to be DS break, so double-stranded breaks, you're completely cutting that chromosome. Okay. DNA repair enzymes, which you will get to learn here in a little bit, recognize this. And they rush in and are wonderful, and they fix everything. But they fix the wrong ones, okay? They take the chunk from this chromosome and they just glue it onto the other chromosome. So they fixed the break. They're recognizing those open ends, right? So they're actually, what they're recognizing is the lack of telomere. Remember we drew out the D-loop when we talked about the ends of chromosomes? One of the reasons those telomeres are on there, one is so you don't lose genetic material every time you undergo replication. But the other way, so it doesn't look like, I always think like bare wires, in electrical. So it doesn't look like bare ends dangling. That DNA repair enzymes are going to see that. They're going to see that bare end hanging and be like, nope, this needs to be fixed. We need to cap that. That needs to be in some sort of way. But instead of sticking the end of our black chromosome on there, they're going to stick this other end that also happened to break. They're quite helpful little enzymes, but they don't always recognize that this is chromosome, number 1, and this is chromosome number 20 or something, So they're not necessarily going to be able to recognize that it's homologous or not. So they're essentially reattaching the wrong chromosome to the wrong end.
So in order for that to happen, 2 different chromosomes have to break.
At the same time, yeah. Yeah, and so that's why, and I'll write a couple of things up here. That's why it's really common in cancer cells where you have a lot of these breakages happening. And in part, this can be due to normal cellular processes, or it can be due to external. And we're going to get into a lot more detail about mutagens and what causes this kind of stuff to happen. But UV irradiation, things like that. So this is big, tend to cause these double-stranded breaks. This would be an environmental cause to the break. And then the repair being wrong, the translocation, that just happens afterwards as your cell's trying to fix this, but it doesn't quite know how to do it perfectly. You also have reactive oxygens, and again, we'll go into more detail when we get into repair, but it doesn't hurt to hear it twice. But you have metabolic, cellular, metabolic byproducts, right? So this would be not environmental, this would be from internal, but O2 radicals. And reactive oxygen types, they tend to make double-stranded breaks in the DNA too. There's tons of others, chemical ways, but these are some of the most common that we see. So your chromosomes do break down over time, this does happen. And there's repair mechanisms in place to fix it, which is great, but occasionally they fix it incorrectly. It's much more common that you would only get one chromosome that breaks, the dangling piece is right here, enzymes can come along, take the dangling piece, stick it back on, and we're good to go. But it's when you have multiple breakages across multiple chromosomes and you just have chunks fall willy-nilly like in the nucleus, and your repair mechanisms are overwhelmed with the work they have to do and they can't quite set it up correctly. So then you see that later as a translocation. Okay, 2 ways. The other way is again involving non-homologous. Okay, so that's kind of the marquee. So they do the normal exchange of DNA, there's a normal recombination. But it's normal when they're homologous chromosomes, it's when non-homologous come together. Okay. So this is actually very rare, but it does occasionally happen where homologous chromosomes, essentially what went wrong here, because the crossover is normal, but what went wrong is their ability to recognize that, oh, you're my buddy number chromosome number 2, we can stand together and have our little crossover thing happen and all that. It's that recognition process that somehow goes awry. So this is, they're actually things called PCs in your cells. They're called pairing centers. They're like, I always picture, someone on a megaphone being like, Julia, stand next to Omar, leave your pair, get together, okay, now you, get together. But you have these proteins, these areas in your cell that are responsible for making the chromosomes come together, okay? So there's zinc finger proteins, there's all these proteins you guys don't need to know, but they help organize cell division. And in particular, they help organize the chromosomal pairs. So they help everyone find each other. And when those proteins are mutated or missing, or other things are going wrong with the cell, essentially, other checkpoints are out of place, then we might see this really rare event happening where non-homologous chromosomes are coming together and they accidentally pair. Honestly, it's amazing to me that any cellular process goes correctly. Once you really think about all the players involved to make all this stuff happen, it's really a miracle that we're all here and semi-awake and functioning. And, there's so much that's involved with things to go correctly. Okay. So those are your 2 ways that you can get translocations. And again, this your book probably I think goes through the inversion loop again. So how we can really see phenotypic consequences when they come together and try to pair up. But you guys aren't responsible for going through all of that for the exam. Any questions about inversions and translocation? Moving to a slightly different, well, it's still on our big picture that we drew of all the chromosomes and the different things that can happen to them. This is in relation to your far right-hand side of that diagram. You had one option was missing chromosome, the other one was a complete extra chromosome. So this is when we have not chunks of material that are deleted or relocated or inverted, but this is when you have an entire chromosome that's duplicated or an entire chromosome that's deleted. So this is variation in chromosome numbers. And some of this you already know. So that's why this chapter should be pretty friendly chapter. So you're familiar with this idea of ploidy. We talked about diploid organisms. Okay. So this group in, when I say ploidy, whenever you're talking about entire ploidy differences, so going from an organism that's normally the wild type would be diploid, if you're moving into, so versus maybe if it becomes triploid, tetraploid, etc. So those would be abnormal if diploid was wild type. Okay. Then these options, that means you're increasing the entire genome, every chromosome. Okay, so every single chromosome has an extra copy of it. And we call anything beyond what is wild type. Okay, so if you're an organism, which you are, that are diploid, and you have an increase to triploid, tetraploid, octoploid, whatever it might be, this is referred to as a whole as multiple, many copies, okay? Some organisms are naturally polyploid, okay? Some, especially plants, have many copies of each chromosome. Some of you guys know I worked, when I was an undergraduate, just like you guys, I worked on sturgeon. And I continue to semi-consult my friends that are still working on sturgeon, and they're a really crazy polyploid, okay? So they, sturgeon, and my friend just found one that's dodecaploid, but for the most part, they're octoploid. But there's also a 12-ploid one, dodeca. So that's the normal, and they're happy, healthy, that's their normal genome state, is to be polyploid. And there's many cells, of course, or many plants, sorry, that are polyploid, insects, reptiles, fish. So my point here is this is normal for some organisms. Not so much for humans and most animals. Okay? Where do we see polyploidy in humans, for example? That's different, and I'll get to that. So trisomy is when you have one chromosome that has too many copies, not every chromosome in the genome. Yeah, but we'll come back to that. Every chromosome, and you guys, everybody in this room right now has a tissue type that is polyploid. Liver. Yeah. So in your liver, you actually have cells that are all different types of ploidy. Actually, there's lots of triploid, some tetraploid cells. So you're sitting around right now with all this extra chromosomes in your liver. Some of you guys have healthy livers. So phenotypically normal livers that are polyploid. So genetically, what would be the advantage to having extra copies of everything? Why would you, Steve?
Possible to increase the expression of certain genes.
So this could, what we've seen is this is evolutionarily, evolved this way so that so it's normal for some organisms. The human liver is an example of a polyploid tissue, okay? And we tend to see these ploidy differences both on a whole organism level and on a tissue level, because maybe more is better in terms of a particular, in that tissue you need to express a certain gene a ton. Maybe there's a ton of gene expression that's going on that has to occur in that particular tissue type. There's all kinds of theories about this, organisms that have evolved to be polyploid as well.